here, metabolism is the driver and genes are just passengers. Isn't that interesting? And this is probably true. Because, and where this came from in his book, he says gene variants can't explain the increase. All these diseases I've just mentioned, they've gone up in incidence in the past, say, 70, 80 years. That can't be a genetic change because it's too quick. So what has changed is the environment, our diet, environmental behavior, that's changed, not the genes. So metabolism, of course, is the thing that's changing in this context. And then the metabolic change is what's driving, if you like, pathologic gene expression. And that's causing disease. So here's metabolism as the driver. Gene expression is just the passenger. And what that means, of course, is understanding this metabolic shift might give us new therapeutic targets is the overall idea. And the big link that NAV makes, of course, that we're all interested in is a metabolic change causes an epigenetic change. So you need acetyl-CoA for acetylation, for instance, be that of histones or DNA. Alpha-ketoglutarate is a key cofactor for the DNA and histone demethylase, remember. And succinate can block them. So again, two very important metabolites. And then carbon-1 metabolism is tied into methylation. So clearly the epigenetic modification that you see that regulates the genes is intimately tied into a metabolic change. And again, we're all trying to unravel this now and get more detail on what that shift might mean. It's very hard to measure these epigenetic changes, at least in my lab. So, you know, but still we need to consider this because it's the metabolic shift that ties into gene expression in a pathologic context. That might be through epigenetics. And that's where the link is, I think, from, you know, metabolism being the driver and gene expression being the passenger. Now, let's get a bit more specific now. So clearly, this business of the mitochondria, if I go back again to that slide for a minute, the mitochondrial change, driving inflammation is the overarching idea. And of course, what this means is the metabolic shift is what was called Warburg, as you all know. Warburg described this, or Warburg for our German friends. Described that first in tumors. He, of course, mainly focused on glycolysis. You might call it aberrant glycolysis, certainly aerobic glycolysis. He missed the mitochondrial dysfunction part, and that is as important as glycolysis for the Warburg effect. Mitochondrial change and disturbance is intimately part of Warburg metabolism in tumors as well. And the tumor guys now are more and more interested actually in targeting, say, complex 1 in the mitochondria, because that becomes deviant in the context of a tumor. So these events for us, though, are driving inflammation is the idea. And what this means as well is mitochondria, getting back to that again, are now front and center. I mean, there's a great review, came out about a year and a half ago now, a couple of years ago now, the rise of mitochondria medicine. And I love this slide. Look at this. This is like battle of the organelles. So the mitochondria took over from the nucleus about 2012, you see. And they became the dominant organelle. This is numbers of papers published. And I'm sure that's a lot to do with the immunologists working on mitochondria. And that began to dominate as the organelle. If you work on the Golgi, you're a loser. You know, it's way down here. So don't work on the Golgi. Switch to mitochondria. That's where the action is. But it just shows you how this intense interest in mitochondria underwent this resurgence, you know, mainly because of cancer, but also the context of immunology and inflammation. And I use this phrase, metabolism trumps genetics, because this is where the action is my overall message. So that's the kind of preamble in a way. Now, that gives you a sense, and I'm sure you're all aware of some of this background anyway, of where the excitement lies, many questions yet to be answered. If you work on the immune system, you have to consider Warburg metabolism now as a key feature and what that might mean for the particular immune cell that you're interested in. Now, I'm going to give you these two stories now. The first is PKM2, which I hope you all know is an enzyme in glycolysis. And what's interesting about this is it has a moonlighting role. It does stuff outside glycolysis. I'll be published this first in 2016 in macrophages, and now our recent paper on TH17. I'm going to give you that story. It was published a couple of months ago. And then, so as Jan said, we've got a massive interest in these two metabolites, succinate and Itaconate, just two of Krebs cycle metabolites. And I use this phrase reborn because suddenly now we've got to understand Krebs cycle in the context of macrophage changes. And that's the second story that I want to give you. So first of all, PKM2. Now, this is a very interesting enzyme. Worked on for a long time by biochemists in the context of glycolysis. And it became clear that there were two forms of PKM2. And obviously it's the last step in glycolysis, peptide pyruvate. But it turns out there's two forms. There's the tetrameric form and the dimeric form. The tetramer is the one that's very active as a pyruvate kinase. That's the one that does its job in glycolysis. But then it can shift into this dimeric form. And people like Matt van der Heijden, various labs have shown in tumors, you get a shift into the dimer. And that now has a different function. It moonlights, non-enzymatic, it goes to the nucleus. And it can do various things in the nucleus. And one thing it regulates is HIF, and that was shown in tumors first, and that's the kind of story I'm going to tell you. So in many ways, this ratio of an enzyme in glycolysis, which is the striking thing, between a tetrameric form and a dimeric form can be governing then for cell behavior. Because if you shift to the dimer, you're going to change gene expression in various ways. And this is only one example. There's many examples of enzymes in glycolysis having this moonlighting role, GAPDH. I'm sure you're probably familiar with some of that literature as well. And we had a paper on that last year as well. So these enzymes in glycolysis are doing stuff outside glycolysis. And this moonlighting role then becomes very, very interesting. And overall, what we found is the dimer is pro-inflammatory and the tetramer is anti, strangely, and that seems to be holding up in various ways. And I'm going to give you a bit of evidence to support that. And our 2016 paper, we began with macrophages. And we showed if you activate macrophages with LPS, or standard inflammatory way to do it, you get dimeric PKM2, goes to the nucleus, interacts with HIF. Lo and behold, all these HIF-dependent genes get turned on by this process. And IL-1 beta, in our work, stood out as a HIF-dependent gene that was PKM2 regulated in this way. And that was interesting. And what was good about this was there were small molecules out there which have been developed by the cancer guys, actually. Because remember, tumors, you often see dimeric PKM2 as well. And there are small molecules that can drive the tetramer. They mimic things like fructose bisphosphate, which is a natural way to do this. And the tetrameric PKM2 is kept out of the nucleus, and therefore you will see an inhibition of IL-1 production because of this tetrameric PKM2, because you've lost the dimer. And that was interesting because we knew then this was now druggable. There was a way to modulate this using small molecules. And my lab began to work on some of these PKM2 modulators. And this piece of data that we published in 2016 was especially interesting, I thought, because we went in vivo and we gave mice the tetramerizers. And they could block IL-1 production if you gave the mice LPS, but they boosted IL-10. So the state of PKM2 seemed to govern whether you get an inflammatory cytokine in the case of IL-1 or an anti-inflammatory one in the case of IL-10. And who would have thought that the oligomeric state of an enzyme in glycolysis would govern the type of cytokine profile you get in vivo? And of course, you want IL-10 because that's going to be anti-inflammatory and protective. Maybe in the context of sepsis, you never know. And that shift tells us something about this phenotypic idea that the phenotype of the macrophage is being governed by metabolism. And it'll make IL-1 if PKM2 is a tetramer. It'll make IL-10 if it's or a dimer rather. It'll make IL-10 if it's a tetramer, and that was an interesting thing at the time. And then our more recent stuff is a postdoc joined my lab from Verona, Stefano Anghiari. He was a T-cell biologist, and he began to work on T cells because PKM2 had been kind of neglected in T cells. There wasn't much on the role of PKM2 in T cells. And using the small molecule, Stefano could get at this question of a role for PKM2. First of all, he showed very good PKM2 activation, just using CD3, CD28 activation, a bog standard T cell activation protocol. One marker of the dimeric PKM2 is phosphorylated PKM2 on serine 37, and that was boosted by using CD3, CD28. You get massive nuclear translocation. It does take a bit of time. It's not a rapid event, but still by 48 hours, all the PKM2 is now nuclear in the activated T cell, and that struck us as interesting. So it did seem to be a signal in T cell activation. And then of course we tried our tetramerizer. TEP46 is the one we use on T cells, and lo and behold, first of all, they could block T cell proliferation very effectively. That was good. Secondly, we began measuring cytokines. They could block TNF production, and T cells are a good source of TNF. And here's an example. Here's the FACS profile for TNF production in T cells. That was again was an anti-inflammatory effect, I guess. And then it got even more interesting when we looked at the T helper subtypes, and TH17s seemed especially sensitive. So you could block TH17 differentiation, big effect there. In fact, you see the quantification here. Certain transcription factors were down as well if you tetramerize PKM2 and TH17. And secondly, TH1 cells, you could block interferon gamma production by tetramerizing PKM2. And again, there's the quantification there. EOMEs, interestingly, seem to be a transcription factor that was especially being regulated here by targeting PKM2. And then it got even more interesting because we noticed that you could block TH17 differentiation or TH1 differentiation, but the T cells became more Treg-like. And of course, regulatory T cells are the anti-inflammatory T cell. You could boost FOXP3, either in unpolarized cells or in TH17 polarized cells, or indeed TH1. We're boosting here. You see the FACS profile. You're boosting the Treg population about threefold if you tetramerize PKM2. So again, that's like a macrophage where you're changing the phenotype of the T cell into a Treg phenotype and away from a Th17 phenotype, just like you've changed the macrophage away from, say, an M1 phenotype into an M2, at least the cytokine profile being more anti-inflammatory in that context. And then, of course, other people have shown that PKM2 was upstream of c-Myc, HIF, and mTOR. And lo and behold, the tetramerizer could block c-Myc induction here, could block HIF, we knew it was involved in HIF, and could also block mTOR. And then we did a big RNA-seq profile, single-cell RNA-seq, and again, the green means repression, and it turned out that a lot of the MYC-dependent genes were down, a lot of the HIF-dependent genes were down, and some of the mTOR genes were down. And over here, we see glycolysis being repressed. Some of the enzymes in glycolysis are here, and again, they were repressed in this way. And the reason for that is that you need dimeretic PKM2 and HIF to regulate glycolysis genes. So you're suppressing glycolysis here in a very effective way too. So this really put PKM2 then as a kind of a key driver of these changes that are happening in the activated T cell. And then we went in vivo with our tetramerizer and a model of autoimmunity demyelination. It's a model of MS is the AE, you're probably all familiar with this. Gave the mice the tetramerizer, and look at this, delayed the onset of clinical features. The mice didn't lose weight, you know. So we see a therapeutic effect now in vivo by tetramerizing PKM2. And remember, this is a Th17 mediated disease or model, I guess. So it's able to suppress the Th17 compartment. It'll be targeting macrophages here as well, of course. And in fact, we did an adoptive transfer experiment just to look at T cells. So ex vivo, we challenged T cells with to drive a Th17 phenotype. We gave them TEP46 ex vivo and then adoptively transferred them, and they are protective. You can see the adoptively transferred T cells are repressing disease here. Mainly, we think because they're more Treg-like. We think ex vivo you generated that Treg phenotype is a possibility. You don't get a full Treg response, to be honest. You can't measure that functionally, so it may not just be that. But certainly, we think part of the mechanism here would be repression of Th17 cells in that context. So the bottom line then is, if you target PKM2, and in particular this moonlighting dimeric function, you're going to see an anti-inflammatory effect. And there's our, I show this when I give this in person. That's the famous metabolic chart. You don't need to know that. That's all you need to know. You get dimeric PKM2, driving HIF, mTOR, MYC, and STAT3. In fact, there was another paper on that recently. And two other labs, as ever in this business, as you all know, you can get scooped. Around the same time we're doing the work, two other labs had very similar data. Claudio Amaro had it, for example. And so this seems to hold up, this pathway, with PKM2 having a key role. Three labs, I guess, have shown it now. And it does seem to be a key regulator of T cell activation in the context of inflammatory T cells. And what's also interesting is this. So there's lots of papers now showing this in a way. Targeting metabolism will force a T cell into a Treg phenotype. So 2DG does it, rapamycin, DMF, which is used for MS, I'm sure you're all aware, dimethyl fumarate, a polyamine metabolism inhibitor, and now TEP46 will push cells towards a Treg phenotype. And that's a fascination because you're targeting metabolic processes here and getting a different T cell subtype from that targeting, I guess is the way to think of it, away from a Th17 cell. So we do think that this, you know, is exciting and for a number of reasons. Obviously, it puts PKM2 in the T cell in the context of glycolysis and metabolic reprogramming, and that's interesting anyway. But secondly, there's a therapeutic potential for targeting PKM2 here. And remember, you're also able to target the macrophage because don't forget the macrophage data being part of this as well. Now, the second story I want to give you is back to Krebs, and I'm going to talk about Itaconate and succinate in various ways, and we've been working on these for a number of years now. You all know about Krebs, I would imagine, because you're all very dedicated students. Remember, remind all of immunologists, it is the central hub of all metabolism. Now, we've all learned this bit, NADH and FADH carrying electrons, the electron transport chain, and that's only one function of Krebs, remember. It may not be the most important function, although obviously it's somewhat important. Equally, you can drive amino acid synthesis from succinyl-CoA. From alpha-KG, you can drive purines and pyrimidines for DNA and RNA. And of course, from citrates, you can drive lipid synthesis. So all the key biomolecules can be made from Krebs cycle. And then remember, you can feed into Krebs as well and burn intermediates, as we all know. Glutamine is a good example of that. So it does four things, Krebs. So it is the central hub of all metabolism. And then, of course, no surprise, we see it changing in macrophage activation, for example, works on. And the two changes I'm going to talk about, obviously succinate, we're working on that now for a number of years, and now itaconate coming off Itaconate. They're the two specific things I want to talk about. Now, itaconate is a very interesting metabolite. It's a good example, I think, of metabolic reprogramming. It's a better definition of that term in a way, because if you don't have any LPS there, you don't see any because Krebs cycle runs. If you give a macrophage LPS, you induce IRG1, that's the enzyme that converts itaconate into itaconate, and you divert away from Krebs. When we talk about metabolic reprogramming, we're inclined to talk about ramping things up and down. You might ramp up OXPHOS or ramp up the pentose phosphatase. This is a bit different. You're diverting away now from metabolic pathway. So it's true metabolic reprogramming, I think, is an example. And then you start making itaconate. And all the evidence suggests it's immunomodulatory. There's evidence for it being anti-inflammatory, as you probably all know, in different contexts. Lots of stuff is coming out on it, describing what it might do. And of course, you know, it is this business of IRG1 diverting itaconate away from Krebs. So it will impact on Krebs cycle because you're skewing Krebs in a sense. And our paper that we published about a year and a half ago now, if you put LPS in a macrophage, you get a massive flux towards this from glutamine for the start, but also from glucose. And we could track using C13 labeling. Everything is pushed into itaconate after, say, 24 hours of LPS. So the macrophage seems to want to break Krebs and divert away into itaconate. And then, of course, the big question became, what does it do? And lots of labs have seen this increase in itaconate now being a very profound effect. It was shown, of course, by Carson Hiller that IRG1 was the enzyme that made it. And there was evidence of itaconate going up even 4 or 5 years ago. People had seen in 1 or 2 papers. So and our work kind of confirmed that and mapped it a bit more. Now, what's also the case is there have been a literature on this going back a good 15 years or more of itaconate being antibacterial. And there was good evidence that can block the glyoxylase shunt, for instance, in various bacterial species. Yersinia pestis is one example of that, actually. TB, it blocks the glyoxylase shunt. So there's some evidence it may be antibacterial directly by inhibiting metabolic pathways in bacteria. You need, some people, this is somewhat controversial because bacteria have ways around this maybe. Maybe in certain circumstances this might work, but overall, the jury's still a bit out on this one, I would think. But then, of course, very relevant to our worries today, it's also antiviral. And we showed in our paper it could block type 1 interferon production. And that'd be useful for a virus to have, say, if a virus drove production of this, you can limit interferon production. There were also 2 papers with direct effects on Zika virus replication. That was in 2019 in the brain. And in fact, they used 4OI in that paper, the derivative that we made, and it protects against Zika-induced brain pathology. So it does seem to have a role in handling Zika virus. And then it can also block influenza virus replication directly, somehow. So that makes it interesting. So there seems to be direct antiviral effects, and there may be effects on bacterial metabolism. And that's one reason maybe why macrophages make it, is the idea here. And we had seen back in 2016 now, I guess, we did a big metabolomic screen, how my lab got into it. We did a huge metabolomic screen on human macrophages, and it stood out as a very abundant metabolite in that metabolomic analysis, much more than lactase. That's the next one, by the way. Now, you need to have LPS there for a long time to see this. Probably 48 hours really, you see this big boost in it. Lactate dominates at other time points, of course. But still, it's a very prominent metabolite in macrophages, human and mouse. It goes up much higher in the mouse than it does in the human, maybe 100-fold higher in mouse. But still, it is elevated in the human system as well. And of course, the question became, what does it do? And my lab began to work on this. Now, very importantly, Max Artyomov had a very interesting observation. Again, I think it was 2016, where itaconate could block SDH. Now, he was following up on some of our work showing that succinate needs to be oxidized through SDH to generate electrons. And you get this reverse electron transport through complex 1 to drive ROS, and ROS can drive HIF. And Max showed that the reason why succinate accumulates in macrophages then is because of itaconate blocking SDH. And he was able to show that was a net anti-inflammatory effect. 2 other labs showed that as well. The lab of Cordes, nice paper on that too. And in fact, it had been shown in 1949, if you can believe it. Itaconate was an SDH inhibitor anyway. So that was kind of known. And Max knew about that, and he was able to link it into macrophages. And then we published a little review, me and Mike Murphy, on this in 2018. that's the effect there, blocking SDH. And then our paper showed it gets out of the mitochondria, activates NRF2, and that has an antioxidant effect. I'll come back to that in a minute. And then Max had another paper showing it could drive ATF3, and that will repress the thing like alpha B zeta, and that'll have an anti-inflammatory effect. So our thing last year on this, 18 rather, was sort of saying Krebs cycle, succinate, itaconate, very interesting effects outside its normal role in the role of Krebs cycle anyway. And then we were able to show as well early on, it is anti-inflammatory. If you give mice itaconate and then give them LPS, you can protect against lethality. This is the standard LPS study model. Give them itaconate, you can extend life in the mice. So that was evidence of an anti-inflammatory effect in vivo. And then we also knocked it down in macrophages. I don't know something this slide, but we knocked down IRG1 using siRNA in human macrophages, and we could boost pro-IL-1 production. There's the knockdown compared to the control. And again, that means you're knocking down an endogenous anti-inflammatory pathway, and then you're boosting IL-1 production. So that was evidence that suggested it was an anti-inflammatory metabolite. And then Mihai Nitsei had a very nice paper in 2019, a year ago now, in sepsis, where in patients who've got this immunoparalysis. Now, it turns out that during sepsis, you do get an immunoparalysis phase. And he was able to show that was IRG1 and itaconate, and he was able to implicate SDH inhibition to some extent in that. And then when he boosted the immune response to relieve this paralysis and sepsis, that lowered IRG1, and beta-glucan was used there. So again, that was more evidence that this pathway would be involved in an anti-inflammatory immunoparalysis effect, I suppose, is the way I would put it. Now, how we got onto the NRF2 part was Dylan, Ryan, and Ivana Mills in my lab, two very talented graduate students. We were discussing it and wondering what was going on. And Dylan made the point that it looks a bit like fumarate structurally. And fumarate had been known to succinate cysteines on target proteins. It can alkylate target proteins. And we wondered then would itaconate, just like fumarate modifies cysteines. And it was kind of just an idea. It may not have worked out. Gave it a go. Lo and behold, it did. It's a very good cysteine modifier. And in our paper, we reported on at least 30 proteins, if not more, that endogenous itaconate can modify. On cysteine, KEAP1 was one of them, and that would drive NRF2, and that would have lots of antioxidant, anti-inflammatory effects. A protein called GILT, it's an antigen presentation, that came as a hit. And LDH, that was a very prominent hit we got actually. Now, we kind of didn't focus on those in the paper, we mainly focused on NRF2, but LDH is a really good target for itaconate. And a thing called annexin-A1, which controls prostaglandin. There's just 4 of them. So my lab is now working on each of these in turn. So there seemed to be a repertoire of proteins, and itaconate could modify cysteines on those proteins. And we show this endogenously. We could show it in IRG1 knockouts that some of these were not getting modified. You know, so we had a good sort of body of evidence to support cysteine modification as a process. And then last year, a paper by Chu Wang came out. He used a different method to measure cytokine or cysteine reactivity rather with itaconate. He made a reagent involving glycosylation, and he got LDH, which was nice. He modified on cysteine 84. He saw GAPDH modification and aldolase. And he found, I think, about 100 proteins in the end, and about half of ours were in his list. So he was able to confirm many of our proteins were being modified in his study as well. And that was a nice thing to see using a different method. You know? And of course, what's also important is DMF, dimethyl fumarate, has been shown to target GAPDH, and that may be part of its anti-inflammatory effect. So in some ways, it's possible that itaconate is the natural form of DMF, actually. That's one possibility. And maybe just by chance then, dimethyl fumarate was invented as a way to mimic a natural anti-inflammatory pathway. And as many of you might know, it is used for MS. It's used by Biogen. So, and then there was a follow-up paper showing that 4OI can target GAPDH as well. So that was quite interesting on cysteine. So what this meant was glycolysis became another aspect of this molecule's anti-inflammatory effects, because it'll repress glycolysis now. And that paper by Wang showed that very clearly, that these are inhibitory effects on these glycolytic enzymes. So now we have glycolysis being blocked. Itaconate, interesting crosstalk there between the mitochondria now and glycolysis, by the way, because it is made in the mitochondria from Krebs and can now regulate glycolysis. I'd like to know what that means in other contexts. So interesting observation, I think. And then the new stuff I want to tell you now, and this is my second story, which I'll give you. We wondered, going back to NLRP3, as I mentioned earlier, would that be a target? And the reason why we hypothesized that was ROS, as I mentioned, is a signal for NLRP3 activation. So Alex Hoofdman in my lab, a very talented graduate student, he wondered this because he said, look, if it drives NRF2, that would boost various antioxidant pathways, and they may lower ROS, and that might impact NLRP3. And that was the initial idea that we had. And so Alex began to test this. And lo and behold, Itaconate, we mainly use the octyl form because that gets across the membrane and gets hydrolyzed, and we have various controls in place to make sure it's the Itaconate that's the active part. Lo and behold, very good inflammasome inhibitor. And you can use ATP to drive NLRP3, and we're measuring IL-1 here. There's Itaconate blocking that. You can use nigericin as a potassium ionophore. People who are in this field will know this inside out. And again, Itaconate very effectively blocks IL-1 there. And then we measured IL-18. That's the other output from this. And again, 4OI could block IL-18 production. So again, we're seeing an inhibition here of NLRP3. And then we also measured pyroptosis, that type of cell death. And again, very nice that we can block pyroptosis with ATP and nigericin. So that's another readout. And then there's a whole load of assays. You can measure ASK. ASK is a sort of a platform protein that forms these specks that NLRP3 assembles on. And you can measure ASK assembly in various assays. And again, Itaconate can block ASK assembly here. This is a cross-linking gel. These are all these ASK oligomers. And again, you can block their formation with octyl Itaconate, which is nice. And then, of course, at this stage, the chance was that it was maybe targeting some other part of the inflammasome, didn't have the NLRP3. So we tested AIM2, a different inflammasome, which has caspase 1 and ASK as well. No effect on AIM2, very nicely. There's 4OI over here, this is AIM2. This is another one, NLRC4, which is another inflammasome that wasn't inhibited. So again, this suggested specificity that Itaconate was targeting NLRP3. And then we got some ex vivo samples from a patient with a thing called CAPS. Now these patients have an activating mutation in NLRP3. They get horrible multi-organ systemic inflammation. They're treated with anti-IL-1 as a standard of care type thing. And lo and behold, CAPS patients will make loads of IL-1 and ex vivo. We could block it with Itaconate. So again, that was evidence for NLRP3 targeting because these patients have an overactive NLRP3. And then we also went in vivo, inevitably. There's a model of gout using uric acid crystals. You get a big into the peritoneum. You get a big IL-1 production. You can block that with Itaconate in vivo. You can block neutrophil accumulation, again, in this in vivo model. And that's NLRP3 dependent. So this was more evidence that NLRP3 is being targeted. And then to get closer to it, Alex set up a reconstitution assay. Now you can reconstitute the inflammasome in 293 cells. You can overexpress all these different components. And then you can activate the pathway when you drive IL-1. And he overexpressed IRG1. And that could repress. And that was nice. And here's, say, P17 being blocked very nicely by overexpression of IRG1. And of course, IRG1, we hope, is making Itaconate in this situation endogenously to repress NLRP3. There's more evidence. Now, this can be a very artificial system. So you've got to take it in combination with the rest of the data. But certainly overexpressing IRG1 is able to repress IL-1. And then we went to the knockouts, of course. We had IRG1 deficient macrophages. We activated the inflammasome. And we can see a boost now in inflammasome activation in the IRG1 knockouts. Again, a lot more caspase 1 P20 compared to control, a lot more P17. So various assays here being boosted in the IRG1 knockout. And again, that was evidence that endogenous Itaconate could block IL-1. And then again, in the knockouts, there was no effect on AIM2. And that was nice as a control here. So there's no boost in the AIM2 inflammasome in the IRG1 knockout. It's only NLRP3. And then, of course, Alex then did the experiment that he was building up to. Was this NRF2 dependent? And guess what? It wasn't. And here we see the evidence. These are NRF2 knockouts. And you can see just as much inhibition with 4OI in the knockout as you can in the wild type. You know, so it's well able to inhibit in the NRF2 knockout. So it doesn't seem to be through NRF2. So we could rule that out as a possible mechanism. The answer was no to that question. So this all then suggested, and given what I've been saying, that Itaconate is a cysteine modifier, could it be modifying one of these components directly? Could it be modifying NLRP3 or NEX7? That's another unique inflammasome component. And we turn to this, and lo and behold, very nicely, we have evidence that a key cysteine NLRP3 is modified by 4OI. It's cysteine 548. And that's in the interface between NLRP3 and NEX7. We've also shown that 4OI can block NEX7 NLRP3 interaction, probably through this cysteine. This is mass spec, so we can identify that cysteine is being modified. And then very nicely, Chu Wang the guy who had the paper in Nature Chemical Biology, we noticed in his supplement, he was able to detect NLRP3 modification. So I got in touch with him and he sent me this nice data. He's a paper about to come out in JACS actually on this. And he could see the same cysteine being modified, CYS548. He found 2 other cysteines, 284 and 720, NLRP being modified by Itaconate. And that was quite neat because it was consistent with our data. So we think this is a cysteine modification event that somehow inhibits assembly of the inflammasome. So now we add NLRP3 to the picture. So not only does Itaconate drive NRF2 in certain contexts, it blocks glycolysis, it inhibits SDH, it drives ATF3, and now it's blocking NLRP3. And for me, this further emphasizes or suggests it's a very important anti-inflammatory pathway, especially what I said earlier, that NLRP3 is a key driver. So many inflammatory pathologies, this seems to be one of the key endogenous brakes on it, we think, that Itaconate can block NLRP3. So we're very excited by that observation. Now to finish, I'd like to talk a bit about human data. So much of what we do is in mouse. It's in mouse macrophages, and you all know the issues with those, especially measuring metabolism for various reasons. And we always worry a little bit about that. We usually use human PBMCs as a complement to the macrophage data. When we do stuff, we always try and replicate it in human monocytes. We go in vivo, which is a better place to go metabolically anyway. So we have various criteria to implicate these things. But we've recently got this data on human with Itaconate, which we're very excited about. Now, this is just beginning, but I've given this in a couple of talks because I'm so excited about it. It was a collaboration with Mihai Netea, who I mentioned earlier. And what this came from was, I refereed this paper, is it about a year ago now maybe, on the structure of IRG1. It's also called ACOD. And it was a very neat structural paper. It's a decarboxylase and all the structural stuff was very nice. In that paper, they reported on variants in the IRG1 gene in humans, which other people seem to have missed. And it turns out that there's at least 6 variants of IRG1 in humans. And one of them, there's an asparagine, is a serine in the variant. It's asparagine 152 serine. This variant was shown in the paper to be 3 times more active than the wild type. Now, this asparagine is in the active site of the serine. It seems to change the KM of the enzyme. So it's a natural activating variant in IRG1. And in fact, it mimics the mouse. The mouse has a serine in that position, which is one reason why maybe in mouse you get more Itaconate. And they talk about this in the paper. But more interestingly for me, they did that genotype in different populations. And they noticed in the African population, there's a 20% frequency in that variant, in this activating variant. It's only 0.2% in the European population. And that got my attention. So that's interesting. Why would this variant, an overactive form of IRG1, be more common in an African population? And you begin to think about things like TB. You begin to think of things like, parasitic infections, maybe Itaconate's protective in some way. And that began to get my attention. And Mihai Netea, I've had great collaborations with over the years with Mihai. I knew that he had challenged PBMCs from African populations and also European populations. And I knew that he'd measured cytokine production. He was doing this as part of an adjuvant vaccine trial. And I said to him, can you go into all your samples? And can you look to see if you can correlate cytokine production in those samples with this variant? Just ask them, because he had all the data, they had thousands of patient samples. And he genotyped them for all different genes. And he had cytokine measurements. And then he sent me this data, and this made me fall off my seat. It's about 3 months ago now when this data came in. It turns out that if you carry that variant in IRG1, the activating variant, you make a lot less interferon gamma, and you make a lot less IL-5. Those 2 cytokines stood out in his analysis. There was a big statistical correlation here. Now, remember, this variant is making more Itaconate, and that Itaconate must be inhibiting interferon gamma and IL-5, those 2 particular cytokines. So for me, this is interesting because it's direct evidence in humans that Itaconate's impacting on 2 very important cytokines. And if you make more Itaconate, because you carry that variant, you make less of these cytokines, and that could be advantageous. Less IL-5 might mean less eosinophilia in a parasitic infection, for instance. Less gamma, of course, could mean less inflammatory macrophage activation. So, and this could give them a survival advantage in the context of infection, say, for instance. So we're looking at this now more closely with Mihai, obviously, and getting more samples and measuring more cytokines and trying to see what this means. But the fact that 20% of the African population are like this must give some survival advantage in the context of infection, we feel. And it validates the pathway in humans, that this is an immunomodulatory pathway. And Itaconate can impact on these cytokines now in humans based on this variant. Of course, there could be other reasons for this. We're just beginning this in a way. But the fact that it's correlating with that variant to me is extremely interesting, given what we know about Itaconate. So again, that's just a little snippet at the end to show the human data. So I'll summarize now and give you the overall picture. So first of all, I told you NLRP3, and of course, it is a metabolic sensor in all these different diseases. Will inhibitors make it? We don't know. Obviously, our company, Inflammasome, now are in the middle of clinical trials, so are many other companies. And remember, many things fail. You know, I think the success rate from a phase 2 trial to a drug is like 1 in 8 or something like that. So still, but are these inhibitors going to make it? I don't know. We'll see. And then the other bit is this bit here. Immunometabolism. Clearly, we think there's lots of opportunities here. New frontier anyway. All these discoveries are fantastic. Who knows what that might tell us, you know, and how you can reprogram cells and so on. Remember, it works already targeting immunometabolism, methotrexate, metformin, DMF. These are drugs in humans already. So we know if you target metabolism, you can see a therapeutic effect. And now, of course, our two big interests, PKM2, will that yield? And now Itaconate. Eat up the multiple things it does. You know, that's now interesting as a prospect. So these two targets, if you like, are interesting. And let's see what happens next in terms of lifting that efficacy ceiling. If we target these things, can we break that ceiling? We might break it through NLRP3 inhibition. We might break it by targeting metabolism. And that's obviously a lot of work to be done to address that. And it may not be true, but let's see what the next phase of all this tells us. And that's in my mind the whole time. Can we translate these discoveries into something useful? Scientifically, in their own right, of course, they're extremely interesting. But now we have potential to really change inflammation and change the immune response in a good way by targeting these different processes. And I'll finish on that and thank all the people. There's Dylan Ryan and Ivana Mills. Dylan's now in Cambridge doing a postdoc with Christian Frezza. Ivana's with Ed Shahani over in Harvard, so they moved on, but they did all the work on Itaconate, the big sort of observation that we made. I told you about the work on NLRP3 with Alex. There he is. And there is Stefano Anghiari, that guy there with the glasses. I've got the wrong name on this. Apologies to Stefano if he's listening in. There's Stefano. He did all the work on PKM2 and T, so it's very important. And then we have our collaborators. Obviously, can't do this without them. They bring ideas, technology, discussions. It's a huge collaboration, as you all know, this business. And Mike Murphy is obviously my key collaborator for years now, and Christian Frezza in Cambridge. Mihai, I told you about the work with him, and Vinod. Richard makes all our reagents from Glasgow. Roman Fisher, he's the big lab that measures cysteine modification. He found the NLRP3 modification. He found the original modifications for us with Itaconate. And some of this was done in GSK with Louise Modus as well. The funding was the European Research Council, the Wellcome, Science Foundation Ireland, and GSK. And I'll finish there and thank you all. I hope you're all still listening.
Thanks a lot, Luke. This was very interesting. And I'm sure in real life, there would be a big applause right now.
I hope so. I can hear it. I can hear the applause.
Something I forgot to mention while starting was that questions could be asked via the chat box, but I'm happy to see that a lot of people found this chat box, and there's some very interesting questions raised there. So I will try to go back into my account, show, and go here. So if there's other questions coming, please ask them via the chat box. I will start maybe in the beginning, where Nidhi Mehta asked whether this TPP46 treatment could have beneficial effects in controlling chronic inflammation in obesity.
That's a great question. Yeah, I mean, TEP46 is a tool compound, of course. It was made at the NCI, so it's not really a drug, you know. It's a strange structure. Now, our company Citrix, by the way, is making better ones. So we'll probably give those out if people want them. But TEP's a good tool compound. But absolutely, I think our work and other labs as well would suggest that in any inflammatory context, you could tetramerize PKM2 and see an anti-inflammatory effect. And certainly in obesity, that will be one to test for definite. It'd be very interesting, for instance, to try insulin resistance in some of those models to see if you can, you know, boost insulin sensitivity. And maybe the effect of glycolysis could be part of that. Because remember, you will soup up the flux through glycolysis with that tetramerizer as well. So that's well worth considering.
And then maybe a question related to this from Jason Hellman. He questions whether these immunosuppressive actions of TPP could also be risky to, for example, enhance the risk for infection. And I could also imagine, for example, in the context of cancer, this is often not the direction you would like to go. So,
yeah, that's a great question. This is always in our minds.
right.
So whenever you tamper with metabolism, you worry about, you know, various unwanted consequences. And whenever you block a key immune pathway, of course, there's always a risk of infection. Now the answer to that is, if you get the dose right, you know, you wouldn't want to turn these things off completely. And we know from methotrexate and from metformin, these are relatively safe drugs and they target metabolism. So I think it's possible to get around the worries to do with, say, immunosuppression through the right dose, I suppose. But still, you need to look at that when anything clinically, you need to be wary of that, or at least be aware, you know, kind of wary of it. It's not so much immunosuppression that worries us, of course. It's just, you know, targeting metabolism in a different tissue that you don't want to target. Like blocking glycolysis may have consequences, who knows, in the gut.
Right.
The hair follicles. I mean, there's evidence for this. Again, there's evidence, though, to suggest that if you can just ramp it down without fully blocking it, normal tissue can compensate. Inflammatory tissue can't. It's a bit like the old idea in tumors where there's addictions to certain metabolic pathways. The same seems to be the case in inflammation. And there was a great paper actually in Nature Medicine on targeting GLUT1 in psoriasis. And that seemed to work. And the psoriatic cells couldn't adapt, but the normal keratinocytes could if you block GLUT1, you know.
Right.
Again, there's enough bits there to suggest that it might not be as toxic as you first might imagine.
And then an interesting question from Louise Ince. She wonders whether LPS stimulation and the effect on Itaconate is also modulated by circadian rhythms in these cells.
That's another great, yeah, Annie Curtis, who's on the list there.
Yeah,
Yeah,
she's on the planning.
Postdocs. She's measuring that, absolutely. I think IRG1 is circadian, actually. Yeah, so
okay.
That'd be really interesting, wouldn't it, to see if it's up, say, during the day when you have an anti-inflammatory state. So that's what I'm considering. And I know Annie's in the middle of doing those kinds of experiments.
Maybe we should ask her again in a few
Yeah,
weeks.
Exactly. Yes, maybe she'll show some data. You never know.
Right. And then Carl Harber, that's a PhD from our lab. He asks, Itaconate obviously inhibits SDH, but he wonders how does succinate then accumulate at these early time points, like 1-2 hours, when Itaconate is actually not there yet. So yeah, how does this kinetics going?
That's a great question as well, as you know, Jan. I mean, it's very dynamic, isn't it? When we did that succinate stuff, glutamine is feeding in through anaplerosis, of course. That's the main source of the succinate. So when you see that elevation, it's just increased flux, really, you know, and more succinate's getting made, and then it's getting metabolized, obviously, as well. So that early succinate is probably coming from glutamine. In our paper, we show that through tracing. And then remember, of course, succinate does two things. We think it goes through SDH, and we get RET and ROS. But then later, it will come out of the mitochondria and stabilize HIF in its own right. So it's a double whammy in a sense, you know. And to be honest, we had trouble implicating SDH inhibition in some of the things we see. I mean, there's evidence for it. I'm not knocking the data, but I don't think SDH is a key target here. I think it's the other things that are happening or may well be more important. You know?
Okay. And then someone with a funny nickname, he or she asks, Itaconate blocks SDH, so again, along this line, which is obviously complex 2 of the electron transport chain. So does this mean it blocks OXPHOS in macrophages?
Well, that's a good question. I mean, of course, as you know, OXPHOS is uncoupled anyway in the macrophage because you get nitrosylation of some of the components, I think in complex 4, for instance. So you don't get much electron transport happening anyway with LPS as a trigger. So that wouldn't really impact then. It's a good question. I think even 30, 40 years ago, it was shown Itaconate could block OXPHOS, though, like in the normal state, because it is an SDH inhibitor. So you would see an effect on OXPHOS. So it's interesting to consider it as an OXPHOS inhibitor, maybe not in the context of LPS in the macrophage, but possibly in other contexts. That's worth thinking about for definite.
What I personally liked a lot was also the link to the human situation and the collaboration you have there with Mihai. Kira, again from our group, asks whether you also looked at IL-1 production there, because obviously that's a very important target of both succinate and Itaconate. So is there something happening? It
was a very unbiased screen, that, to be honest. I didn't say to him to look for things. I'd love to have seen IL-1 come up, wouldn't I? I said, just look at all that, just come back to me. Is there any difference? Now, he measures hundreds of cytokines in that assay, you know. And then he came back with those. He said those two were the most statistically strong thing. But he did say other ones were in there that didn't reach statistical significance. So we're going to go back now, of course, and get more patient samples and maybe look for significance. But we should see an effect on pro-IL-1. That would be expected, I suppose. Now, it's a funny assay because it's PBMCs. It's not like macrophages isolated, you know, so you're looking at a bit of a mix of cell types there. So, but now, of course, the window's open to try different cell types, see differences there, hopefully. So we're hopefully going to see changes. If we don't, of course, it'll go against our overall idea, which wouldn't be too nice. But let's just see what the data tells us.
Yeah.
We're intrigued by the IL-5 gamma because obviously there could be a role in eosinophils now in humans. You never know, you know. So we're looking for IL-5 anyway. So we're looking more and more at that kind of aspect from that data as well.
Okay, and then maybe a last question. I think we have to somehow conclude somewhere. It's from Lord Vader. He's also watching. So he asks whether there is evidence of Itaconate having effects in other cell types, fibroblasts, and what would be the impact?
Yeah, well, that's a great question as well. So the Zika virus paper I mentioned, that was all in neurons. So neurons can make it and can modulate Zika inside the neuron. So there's one cell type that it's made in. IRG1 is very restricted in expression. It's mainly in macrophages, myeloid cells. You don't see much in fibroblasts, so you wouldn't see effects on other cells. But the other thing we're very interested in is, does Itaconate get into other cells? So you might have a situation where the macrophage releases it, either through cell death or maybe in an active process. And people have measured Itaconate extracellularly in inflamed tissues and so on. So you do see it going up. So there could be a transporter. It could be a receptor, like succinate has a receptor. So that means it could impact on other cell types. And that's a question that we're very interested in.
Yeah, very interesting. These are also things that probably everybody's wondering. What's the GPR for Itaconate? Are there transporters? This kind of
We're looking for that as we speak. Well, we can't do any. I don't know about you guys. Our labs are shut. We can't go into the lab.
Same here.
Hauke Weiss in my lab, that's his project. He's looking for the transporter and the receptor as we speak.
Okay, wow. Okay.
Screens and so on, try and find those. I bet there will be a receptor, and it'll be like the succinate receptor, you know, and there's bound to be a transporter. There was a paper actually showing M2 macrophages take it up a lot. They really can accumulate it. So there must be, I think there's a transporter in M2s maybe. Yeah. You wonder what that means, you know, what's it doing to an M2 macrophage. So we're very interested in that as well.
Okay. Yeah, looking forward to this new data. I think we have to conclude here. Thanks a lot, Luke. This was really nice, and I'm happy that you accepted to be the first speaker here. I really look forward to this next week's. Some household things. There was another question asking, will the recording be available afterwards? I promised I will ask all speakers. Luke agreed, so thank you for that. We will make it available. Please look at immunometnet.com or at Immunometnet or at Immunometlab on Twitter. We will make all the links available there. We will update the programs. Obviously, there's other people organizing very interesting seminars. So I mentioned a few. Take a look at them if you have time or if you are bored. And in the end, Immunometnet was meant to be a physical symposium organized here in Amsterdam. We were sponsored and they made this possible. And in the end, yeah, Immunometnet is the reason we're all talking together here now. So if you have time, take a look at the sponsors and their products. They have quite some interesting things also for immunometabolism research. So I thank you all. I look forward to see you again next week. Then David Finley will present. He's Luke's neighbor in Trinity College. And I guess we will focus there on the NK cells in relation to immunometabolism. So thank you all, and hope to see you next week.
Great. Thanks very much, and thanks for organizing it. It was a real pleasure. Thanks a lot.
Bye, Luke.